Prayer is the song of heart that reaches the ear of God. With this thought, I request the student of Department of Zoology to sing the prayer song. Let us all remain standing for the prayer. God's blessing. I now invite J. John J. Kamara, chaplain, to lead us in prayer.
Only gracious God, we thy children come to your presence, O Lord. Praise you and thank you for this blessed day you've added in our lives. Thank you, Lord, for the wonderful program, the Endowment Series lecture in honor of Professor JCBA Endowment. God bless the department, the heads of the department, and the faculty members who toil for this program. Let it be a blessing to all the faculty members and the student friends gathered here, O Lord. Thank you for our management, our principal and secretary, for the wonderful encouragement and support. Especially praise you and thank you, Lord, for the presence of Dr. Kanagaraj, the special resource person of this day program, O Lord. We commit him into your presence. Bless him, O Lord, bless all his responsibility and all his contributions to the field he associated with. We commit every one of us gathered here into your presence. Be with us and guide us with your presence. We ask this prayer in the mighty name of Lord and Savior Jesus Christ. Amen.
A word of welcome to all the distinguished guests on the stage, off the stage. It's a privilege for the Postgraduate and Research Department of Zoology to organize Professor JCB Abraham Endowment Lecture Series 2. And this is the brainchild of Professor Dinagaran Michael, former head of the Department of Zoology, and Professor JCBA Abraham students. The present day students will be wondering about who is Professor JCB Abraham. Anyone of you know who is Professor JCB Abraham? Yes, he was a professor. Yes. He was the former head of the undergraduate and postgraduate head of the Department of Zoology. He has inspired many students by his lecture. Professor JCBA loved and taught evolution and genetics. In memory of Professor JCBA and his love for subjects like genetics and evolution, this endowment lecture blossomed. So just like you students, few students who have gone out of this college, they have started this endowment lecture. They have collected a certain amount of money. They have made it as an FD, and out of this interest, this lecture is being conducted. And this is in honor of a professor. And to address this lecture, we have a renowned scientist, Dr. K. Thangaraj, who is a Bhatnagar Fellow. This is an award given to outstanding scientists in the country. He has a very extensive expertise in ancient DNA and forensic genetics. His research interest in the field of human origin, health and disease. He is a world-renowned scientist in human genetics. He is the former director, Center for DNA Fingerprinting and Diagnostics, Hyderabad. His research papers have high H-index. He has received many awards, and one such recent award is a legend. He's from Hyderabad, and he is given the award Legend of Hyderabad by Dr. Mohan Diabetes Specialty Center. Give him a big hand. On behalf of the Postgraduate and Research Department of Zoology, I welcome you, sir. It is an honor to have you with us for the JCBA Endowment Lecture. Welcome. It is my honor to welcome our former head of the Department of Zoology, Dr. Dinakaran Michael, who has joined with us online. He is a versatile person, an administrator, an active researcher. Of all, he has a love for the department and the college, which has bloomed into the lecture series. Welcome, sir. He is joining us online. It is my esteemed honor to welcome our esteemed principal, Dr. Paul Jacob, a pillar of support to all the departmental programs, who is paving way to take this institution to greater heights. Welcome, sir. It is my honor to welcome our Vice Principal, Dr. Samuel Anduselvan, who encourages us and spins all the activities in the college. Welcome, sir. I welcome our Bursar, Dr. Beulah Ruby Kamalam, who is a very, very powerful person, like Nirmala Sitaraman. She is very supportive to all the activities in the Department of Zoology. Welcome, ma'am. I welcome our chaplain, who is the religious head of this college. Welcome you, sir. I welcome all our alumni who have joined both online and offline to participate in this lecture series. A special word of welcome to all the donors of JCBA Endowment who have joined online. I welcome our esteemed alumni, Mr. Prince, who's a retired master from Sainik School, who's always there for the department, an ardent lover of JCBA. I welcome you, sir, on behalf of the department. And he's the very youngest alumni. Can you see him? He's very young, energetic still. And I welcome all faculty friends of this college and other colleges. I welcome our former head of the department, Dr. Joseph Tatayas, all the heads of the other departments, deans, and I welcome all my dear students from this college and other colleges who are going to be the stakeholders, and my special word of welcome to all my colleagues. Without their support, this program would not have been possible. Thank you, one and all.
Thank you, ma'am, for your warm words of welcome.
It's a pleasure to have with us Professor Dr. R. Dinakaran Michael, former Head Department of Zoology, the American College, former Dean of Life Science, Wales Institute of Science and Technology, Chennai, to address the gathering and share the vision and purpose of this endowment lecture series.
Especially by the science departments, because definitely we'll have at least a person or more than one from the IITs and IIScs, or also we'll have scientists. And our college is known for a few Bhatnagar awardees, and here we have Dr. Thangaraj. He is a CSIR Bhatnagar fellow, and it is a great joy to have him here to talk to young minds, because I always believe that talking to our students, especially great scientists like Dr. Thangaraj and others, that will instill quite a lot of interest among students. And I have always believed that anything which happens outside the classroom, a talk or a discussion or a conversation with such reputed, academicians, which will always bring in a lot of interest among students, and that is what I want actually. So I've been telling my students that in early 80s and late 80s and early 90s, we used to send at least a dozen students to IITs and IISs. That has come down a little bit. But then I've been talking to students, especially the sciences. Whenever I meet them, I ask them, are you ready to take up these entrance tests? And Department of Physics also conducted awareness program on IITs and IISs. And we had one of our alumni from the IIT Chennai to talk to our students. So friends, I would like to again begin by extending my heartfelt greetings to our distinguished resource person, Dr. Tangaraj, CSIR Bhatnagar Fellow, CSIR Center for Cellular and Molecular Biology, Hyderabad. As I was also talking to him about Hyderabad, I do not know how many of you students have gone to Hyderabad, but I tell you, travel is also another type of education. If you go to Hyderabad, probably you'll get the shock of your life. But most of us only know the old Hyderabad, but the new Hyderabad is full of skyscrapers, a lot of opportunities for young minds. So I want all of you to travel to cosmopoles or the metropoles where you have challenges, but if you do well in your studies, you get an opportunity to study in one of the premier institutions in India, and then sky is the limit for you. So, and also I'm very happy to welcome and with respect Dr. Dinaran Michael, former head of the department, and he is also another person who is a great enthusiast as far as academia is concerned. When he started the microbiology department in 1997, I was given the opportunity to take English class. So Professor Nedamaran and I used to go to his department. After taking class, he would give us a cup of tea. Even the interaction with them will always be something related to academia. And therefore, you have such brains which would always give all young minds that you should work hard and then go up the ladder as far as the academia is concerned. So the theme of today's lecture, Who Are We? A Tale Behind Tracing Our Roots Beyond, is both very intriguing and I would say also very profound. From the standpoint of a student of literature, this question echoes through the pages of human history and the creativity. Therefore, the search for identity, be it biological, cultural, or linguistic, has always been central to our understanding of who we are. In literature, in our field of study, this quest often manifests as a journey of self-discovery. In science, it becomes an empirical pursuit to trace our genetic and evolutionary roots. Both seek the same truth through different lenses. So the mystery of human origin and the meaning of existence. So I see a great connect between your field of study and our field of study. There's always something which is very common. And therefore I think and believe that this lecture on Who Are We? A Tale Behind Tracing Our Roots and Beyond is going to be very interesting, at the same time going to pose a lot of questions to you, for which if you are able to find answers, definitely one day you will be one of the greatest scientists that the India produces every year. So as I told you already, lectures of these kinds are very, very important as far as I'm concerned. You always hear lectures, listen to lectures. Sometimes, most of the time, it's just hearing, right? Students? Do you listen to lectures or do you hear lectures? Most of the time, you hear lectures. You don't listen to that. But there's a lot of difference between hearing and listening. But I want you to, concentrate and listen to the lectures that are given to you. Each lecture will have just a spark of idea. And if you are intelligent enough, you'll be able to get that spark. And that would really help you to do quite a lot of things. Today you would be there. And tomorrow you would be in some part of the world. A lot of alumni are spread throughout the globe. And you have lots and lots of alumni throughout the world. And all those people are inspiration to you. So I want, again, all of you to keenly listen to the lecture. Listen in capitals, okay? Listen to the lectures. One way of, listening very carefully is taking notes. I'm happy that many of you have come with the note and the pen. And please listen and take notes. And I want you to develop ideas from here, because this is where ideas develop. Like in our field, we say only in seminars and conferences, theories evolve, language theories. Likewise, I believe that a lot of ideas would evolve, and you take that idea and then work on it. You can write a paper, of course, with the help of your teachers. And therefore, I wish all the best to all of you, especially the students, that you'll have one of the best times for this month and before you go for examination. Definitely there is going to be some test as far as your intelligence is concerned. And with those few words, I wish all of you a great success for this special lecture. Thank you.
Thank you, sir, for your motivating and insightful words.
Respected Principal Dr. Paul Jacob, revered Speaker Dr. Tangaraj, able conveners of the meeting Dr. Joy Sharmila and Dr. Madurai, Head of Microbiology Dr. Joseph Tatoyus, worthy faculty fellows, dear students and ladies and gentlemen. At the outset, I thank the organizers for giving me the opportunity for speaking a few words about this meeting. The main purpose of this series is to give an opportunity to students to listen to eminent scientists, biologists of this country. And this meeting is sponsored by the endowment established in memory and honor of Professor Dr. JCBA Abraham, the teacher par excellence. Dr. JCBA, he was born in October 1927, and also he left us in October 2002. In fact, in the next 2 years, we are going to celebrate Dr. JCBA's centenary year, 2027. And you will have a fairly big gala meeting on that day here in this department. Professor Bhaskar Abraham, that is J.C. Bhaskar Abraham, he did his bachelor's program in the American College, and then for master's he went to Madras Christian College. At that time, American College did not have master's program in zoology. After finishing his master's in Madras Christian College, he joined American College in the early 1950s. In the next 10 to 12 years, he became the head of undergraduate zoology. And then in another 10 years or 12 years, he became the head of the postgraduate department, in addition to becoming the vice principal of the college. As a student, he was very outstanding here, both in American College and Madras Christian College. And as a faculty member in the American College, he was excellent in his teaching. And he inspired large number of students, including myself, and his lectures were wonderful, particularly on genetics and evolution. And I remember in the second BSc, he taught us evolution of the vertebrates, mainly written by Dr. Colbert. That was very fascinating lectures. And then in 1986, he retired from the American College in the as the postgraduate head. But the department, we were there at that time. In fact, I took charge from him, and we invited him after his retirement for nearly 14 years as a visiting professor to the postgraduate department of zoology. And every week he will come and give 2 hours or 3 hours of lecture to the MSc students. So, in fact, the next generation students are also kind of had the opportunity of listening to Professor Bhaskar Abraham. Now, after his retirement, nearly 20 years after his retirement, some of his students thought that we should establish an endowment in honor and in memory of Professor J.C. Bhaskar Abraham or JCBA Abraham or just JCBA. And so we are here in the second of the series of lectures sponsored by the endowment established a couple of years back. And because he was born in October and also left us in October, different years, about 74 years difference, and you expect lectures from eminent people of this country during the time September, October, unless there is any other important meeting in the department, we will always have it in October. So you can expect a meeting in October every year to be given by different experts. And this is the main point I want to tell you about Professor Bhaskar Abraham. And this endowment is in his honor, and in the endowment is supporting these lectures given by the different experts. I don't want to extend, and because there is a time crunch, and so I just finish it by telling, as the convener of the endowment committee, I extend a very warm welcome to all the dignitaries and also to the participants, and also wish them a great time listening to the wonderful presentation by Dr. Tangaraj. Thank you.
I'm the oldest student who has come here of Professor JCBA. Before going into my memories of JCBA, a word about today's chief guest and speaker. Sir, I was going through your biodata some 3 days back. I read and read, I could not even reading it, I became so breathless. And so many responsibilities you are carrying on your shoulder, so many duties, and so much of research work. And above all, your scientific research that you are doing now. And such a great, eminent scientist today have come to honor this lecture series. So for that I thank you, sir. Now coming to my professor, JCBA or J.C. Bhaskar Abraham. It's not today or yesterday or day before. I was the student 64 years back. 64 years. And, but even today, I can see his face as if he is talking to us and he is present in this room so closely and listening to me. What can I say of Professor JCBA? I'll borrow the style of the love stories. First line. I don't know how many of you have read that love story. This is how it goes. What can I say of a professor who is exemplary in his teaching ways? What can I say of a professor who read all the books, fiction, non-fiction, academic, non-academic? What can I say of a professor who is a lover of music? What can I say of a professor who is lover of cricket? And what can I say of a professor that strict disciplinary? And finally, what can I say of a professor that died young at the age of 75? Only the last part is slightly saddening. Otherwise, my professor, our professor, he still lives with us. About his teaching, I did not tell you. Everybody would have talked. Last year also we spoke about that. This year also, and people will talk about that. So I would like to keep myself away from the four walls of the classroom, outside the classroom. Man of quick wit. You ask him anything. As soon as the class is over, some 4 or 5 will follow him. From the sofa hall, will come down, surround him, ask him anything and everything. You know, he is a bachelor all his life. Once we asked him, sir, your idol is a man who married 4 times. But why you are not married? So his immediate answer would be, my idol has not realized his past mistake, even of the 4 times. But I have taken a lesson from him. That's why I stay a bachelor all these years. Another time we asked him, sir, your name is Abraham. What's the difference between the biblical Abraham and you? Instant retort. The biblical Abraham is the father of faith, whereas I am not even a father of children. That's because he has not yet married. So his quick wit and quick answer, that's good. Second thing about his books, he will not lend his book to us, but he will make every one of the students to buy his own book. He will recommend a nice book. So let me see. Don't touch it, man. You go and buy your own. So he made us all buy books. So the few books which I have is because of his recommendation. And over and above that, after some 15 days he will ask the content of the book, what you have read from the book. The same way with cricket. He is not just like a cricket fan like you and me, just doing the score, whether who has won, who has lost. He knew the history of cricket from the beginning. Professor Vasantan has written an article on him, former professor of American College. In that he mentions he once went to Lord's, and in the Lord's it is not a displaying time, it is an ordinary time. So there he went to Lord's Museum, and there were many who were there as spectators. So they were watching, and the curator there was telling that this is the bat used by Grace and this is the bat used by Don Brackman, like that. So he started to talk with him, and he knew more than the curator of that museum. So people, instead of going and asking the curator things, they started following him. He is going around and people are following him. So his cricket knowledge, you cannot just compare. I think it's in the year 1967. It's a match between West Indies and India, and it's a Pongal holiday, and he will always go to Chennai to watch the match. You and I may not spend money to go on to see a test match, but he will go from here to reserve his ticket during Pongal holidays, and during those days the matches were for 6 days. So 1, 2, 3, and fourth day will be a rest day, and then fifth and sixth. So he will see the first 3 days, and rest day he will come here. He will come and ask the students, what the score man today? If you don't tell, he will fire us. If we told, Prachiko, like that. So he is so interested in cricket. Of course I can keep on telling him. Now I came through the main hall, I came through the chapel, I came through the Hampton, of course we are in the Hampton Hall. I can see him everywhere. He is a sound atheist. He is a non-believer. But he will come to Vespers every Sunday. If you ask him, sir, why you are here? I came to see my friends, man. His friends were Dr. Gunraj and Dr. Harris. So he will he will sit at the last row and listen and come. Then when he meets us, what, did God speak to you? He will ask, no sir, but we heard the chaplain talk. So the chaplain is your God? No sir. So he will make everything lightly and without any fuss. He will tell everybody that he is a non-believer. That is a story which goes. My friends went to see him in the hospital when he was terminally ill, probably 2 or 3 days. So after meeting him, they do not know what to say. So they simply told, okay sir, we are going, we will pray for you. He told, no, don't do it. So even during his terminal days, last days, he was so strong, a non-believer in God. Such a beautiful man he is. Stood for his principles. And today, there's going to be a lecture on him. Madam Joy has given me only 3 minutes. I have exceeded it by more than 5 minutes. Hope she is not having a stopwatch with her. And the final bottom line is, we, his student, loved him and he loved us. Thank you.
Now I call Dr. T. Angelin, Assistant Professor, Department of Zoology, to present the memento to our
listening our lecture of our eminent scientist.
I have the photograph of our Professor Dr. JCBA Abraham, who was with me. I was the secretary, PG secretary of the students. So it was the photograph in 1977. Imagine how he is young. You can go ahead when you are going out or your team break. Thank you so much.
Invite Dr. J. Stella Mary, Assistant Professor, Department of Zoology, to greet our alumni, Dr. Vaniyarajan.
It gives me a great pleasure in introducing our chief guest today, Dr. Thangaraj. So, thank you for accepting and coming here, sir. And Dr. Thangaraj is presently CSI Bhatnagar Fellow in the Center for Cellular and Molecular Biology, Hyderabad. And he was former director for CDFD, the Center for DNA Fingerprinting and Diagnostics in Hyderabad between 2020 and 2023. And during February 2022 and August 2022, he held the position of Director Additional Charge of the Institution for Stem Cell Science and Regenerative Medicine, INSTEM, in Bengaluru. And his main research interest for the last 30 years has been in the area of population and medical genomics. Yeah, it's a very great field. And has more than 300 publications in almost all top scientific journals. Few to name are Science, Cell, Lancet, Nature, Nature Genetics. These are very few. He has given very few only. Okay. So then he was the elected fellow of Indian National Science Academy, Indian Academy of Science, and the National Academy of Sciences. It's a great privilege to be a fellow of any one of the academies. He has been elected as a fellow in all the three. And he has received several awards, very few here I am mentioning. It's J.C. Bose Fellowship, Sun Pharma Research Award in Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, and Distinguished Scientist Award, Sir C. Raman Memorial Lecture Award, Sir Dr. Even Brahmachari Award, and so on. And he has delivered several lectures, which of course has earned him these fellowships and awards. And he happens to be the board editor for the journal, the leading journal, the Mitochondrion, and he happens to be the associate editor of BMC Medical Genomics, Tropical Medicine and International Health, and he happens to be a member of editorial board of Human Genetics and Clinical Genetics. And he was the president of the Indian Society of Human Genomics between 2011 and 2015. And he has founded a society, and he happens to be the president of that society called the Society for Mitochondrial Research and Medicine. So these are very few, very teeny tiny bits of his achievements which I have mentioned here. So now it is my great pleasure to welcome our chief guest, speaker today, to make us understand who really we are through his lecture. So the floor is yours, sir.
J.C. Abraham's former students and reading about him, we came to know that how passionate Professor J.C.B. Abraham in the field of evolutionary biology or evolution. And also how much he has loved the subject, students, the way of teaching and so on. I think they had an excellent mentor, helped them in learning and achieve their own scientific way. So it's indeed my pleasure to deliver Professor JCB Abraham Endowment Lecture. The title of my talk, as you all see, I think it's very close to heart of Professor JCBA Abraham. And if you go out of this country or out of the state, say that I belong to Tamil Nadu. I am Indian, right? But if you trace our root, where we have come from, that's what I'm going to talk to you. And studying the evolution, how to be able to understand even more which you would not have thought. Right? So both aspects which I'm going to cover in this talk. If you look at this picture, in India alone, we have 1.4 billion people, right, with a great amount of diversity. That question is very, very important. As an evolutionary biologist or a student of zoology who study evolution, and we all believe that we, the human and chimpanzee, have diverged from a common ancestor called hominids. This is very well established. How can we say that we are close to chimpanzee? If you compare our genome, we share 98% of our genome with chimpanzee. Even lower vertebrate or invertebrate or even plant genome, if you look at, we have some proportion of genome which we share with many organisms. That shows that how the evolution has took place for several millions of years. And I also want to show you a slide because I read Professor JCBA Abraham was a student or at least spent some time with one of the evolutionary biologists, Jansky. Right? And one of his articles, which was published in the Scientific Monthly, the title, I don't know whether you can read, was The Raw Material of Evolution. So what is the raw material of evolution? Right? Mutation. So without genetic mutation, there is no evolution. Right? That means the genetics play a major role in the evolution. The DNA change brings so much of phenotypic difference. In this article, he mentioned that in nearly 80 years of evolution, it's very difficult to say, but I can say that it has been a very difficult time for the 80 years which he mentioned. Because that 80 years which he mentioned is, you can see 1938. The one which mentioned 80 years is Charles Darwin's book, where he wrote a book called The Origin of Species. So that's where he for the first time, based on the anatomical similarity, showed that we are close to chimpanzee or apes and so on, and gave kind of thought that we have evolved from non-human primates. Do you all believe in evolution or not? Or we have evolved from non-human primate, or we are close to chimpanzee and other non-human primate, right? And the genetics is behind that, the evolution. Right? There's anybody who don't believe on evolution? You don't believe on evolution? Okay? I'll give you some example. Look at these three brothers who are born to a normal parents. a change in our phenotype. Probably there is a master regulatory gene or mutation which we all gained during the evolution. There was a back mutation. Therefore, these three brothers who are born to a normal brother have chimpanzee-like features. This is one example. Second example, you see this man and his daughter. One of the features in the human evolution is losing of air on our body. Again, there could be a mutation which helped in that phenotype. Probably there's a back mutation which has brought the air on the body full of air on this child. The third example is for Dr. Joyce to see, because he is not believing on evolution. Another feature which we gain during the evolution is walking with two legs. It's called as bipedalism. Right? And this picture, what you see, three sisters. In fact, there was a 5 children came born to your normal parents. As a Turkish family. If somebody want to see more, you can see the video where the 4 sisters can walk with only 4. They cannot walk with 2 legs in this posture what we are today. Only one brother, the elder brother, can walk with 2 legs, but not steadily, as a drunkard. Okay. There's no proper balance. So these are all some of the features which we gain during the evolution. Even single base mutation out of 3 billion base pair can change so much of drastic phenotype. One mutation, the gene called SRY, sex determining region on the Y chromosome, can change male to female phenotype despite the presence of Y chromosome. Similarly, androgen receptor gene, just one single base mutation can change male to female phenotype. So again, reiterating that genetics is played a role in evolution. And this skull, if you see, both human and chimpanzee, is again another feature is increase of our brain size. There's again gene for that. Of course, the gene is conserved, myosin heavy chain. The mutation which we have, if you compare the sequence of human and chimpanzee, there's a difference. So that difference created more space in the skull. Therefore, the brain sizes increased. Similarly, there are few more genes. There's another gene called protonorephin. That gene, or the mutation in that gene, that actually helped the human for learning, experience of pain, perception, behavior and memory. These are some of the unique features of human. Right? And there's another gene called FOXP2 that is essential for human speech and language. The mutation which created helped in making the muscle loose, therefore the human able to talk or communicate. So again I am coming back to this slide, reiterating that human and chimpanzee have diverged from a common ancestor called hominids. Not today or yesterday, it was about 6 to 7 billion years ago. And these are 6 to 7 million years. There are several species. For better reading, I just split that into 2 columns. You can see these are 7 million years. So in this process, there are several species have evolved and they are all disappeared. There are 3 distinct features which we gain. One, the brain size which I already spoke, walking with two legs, again that I demonstrated. Third one is the ability to make tools for the survival. Animal doesn't use the tools, but human develop tool for survival. So I already mentioned about brain size. You can see the brain size, the chimpanzee or the early species chimpanzees, it was as similar as chimpanzee. It has increased to 35% in Australopithecus, then increased to 50% in Homo habilis. Right, you can see the cursor, right? Then 60% Castor, and gradually increased. And our close species in terms of evolution, Neanderthals, it was predicted that has slightly bigger brain than the human. But we don't know whether they were intelligent than human or not. But why this species has vanished? Later it was found that human and Neanderthals have admixed. Interpret that I will come bit later. And the reason for why this species vanished, there are several theories. One of the interesting theory proposed by Paul Millers from Cambridge, that he said that the inherent violent behavior of modern human probably responsible for vanishing of this species. Right? We all know how violent we are. Right? That could be one of the reasons. So coming to this species, Homo neanderthals, it was found in Europe. These are the place where the Neanderthal skulls were found. In the recent past, several extensive work has been done, particularly by Samantha Pavo. He was the one who sequenced the bones or fossils using ancient DNA technology. And he has shown the link between the human and the rest of the other Homo species. And because of his work, he has been awarded the Nobel Prize for Medicine and Physiology about 2 years back. Then if one can ask a question, if you say the evolution, and Neanderthal is our close relative in terms of evolution, whether we share some amount of genome or DNA. This is some time ago, the year 2016, we along with several scientists around the world, the study was led by one of my friends, David H. from Harvard Medical School, where I was part of it and Samantha Pavo was part of it. And we have shown that the people who have migrated out of Africa all carry up to 3% of Neanderthal genome. We all carry the Neanderthal genome up to 3%. We also carry the Denisovan, this is another one which I am not covering here, up to 0.5%. Okay, that shows, and in the recent past there are several other studies which again conducted by subject matter. Now let me come to origin of modern human. And who is the first modern human who migrated out of Africa? And it's well established that the modern human arose in Africa about 160,000 years back and migrated out of Africa and populated all over the world. And who is the one who migrated out of Africa? I don't know anybody of anyone of you are aware about this particular population. Any idea who are they? But Homo sapiens, but where are they? Any guess? Sorry? Africa, They are in Andaman and Nicobar Islands. Sentinel, yeah, Onge, Jarawa, Sentinelese and Great Andaman. There are 4 tribal groups. And we had an opportunity several years back to go and collect sample from Andaman and Nicobar Islands and sequence the mitochondrial genome of the Andamanis. Of course, we have been studying for many more people. When we sequence the complete mitochondrial DNA, the reason is, of course, we use different set of markers for understanding human origin, migration, and so on. So one of the markers is the mitochondrial DNA because of the inheritance of mitochondria, which comes only from the mother. We all inherit the mitochondria and mitochondrial DNA only from the mother. There is no paternal contribution. There's a paternal contribution always leads to some kind of disease. So mitochondria also known as the powerhouse of the cell, right? So you know that where we all get the power from? Get from mother. Okay. So looking at the mutation in the mitochondrial DNA, we can construct different haplogroups. The worldwide population mitochondrial DNA haplogroups have been well established. I'm not going into technical aspect. I know that it's a very diverse audience. And based on that, I'm just giving only the conclusion. Based on that, we concluded that the Andamanis are the first modern human who have migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. Now we can imagine that just looking at the mitochondrial DNA sequence variations, what kind of conclusion one can draw. But so far there's no discussion about this, whether is there any alternate way of migration. Okay, this is the first wave of migration which people have come from out of Africa. The Andamanis are the first modern human who have migrated out of Africa. Subsequently, we studied several other populations, but now I am coming to the other important study which we did. After the human genome was sequenced, variations which are present in the nuclear genes or nuclear genome have been identified and has been available several years ago, about 1 million genetic variations across the genome, and we used that 1 million genetic variations and reconstructed the Indian population history. So what we have shown at that time was this. I showed you that the Andamanis are the first modern human who migrated out of Africa. That's a deep-rooted divergence. And during that migration, some groups stayed in southern part of India, whom we call as ancestral South Indians. Okay, so that migration brought the people. That means South Indians, either Kerala or Tamil Nadu, is the first modern human who have settled out of Africa migration. Then there was second wave of migration for people. That means from Africa, they moved towards north. Somewhere in the Middle East, there was a divergence. One group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North India. There are several evidence. Subsequently, we have shown the mutation which turned the skin color into lighter is present in Europe, Middle East and northern part of India, but not in southern part. So that's one example. The lactate persistent gene mutation. We have lactate persistent mutation, that the mutation that helps us for better digestion of the sugar which is present in the milk. Still today, there are few who cannot digest the milk. They drink milk or curd or any milk product, they get stomach upset, vomit, loose motion and so on. That's because the enzyme which digests milk sugar is active only during the weaning period. After that it won't be active. Because of the domestication of animal in the Middle East, people started drinking the milk. They got the mutation which helps in digesting the milk sugar. Okay, that mutation is common in Middle East, Europe and northern part of India, again not in southern part of India. There are several other mutations which gives connection between Europe and Middle East as well as northern part of India. Okay, that's connection. And this part shall come a bit later, that ancestral South Indian and ancestral North Indians have admixed. Right? That admixture you can clearly see here. There are several individuals in the PCS called as principal component analysis. Europeans, Africans and Chinese. Several individuals in a single cluster. I mean, there's no difference at genetic level, therefore they are in a single cluster. Whereas look at Indian, there are several, but they are not in single cluster. Each population is one cluster. That's one. Second, although each population is unique, but each one of us have some kind of genetic affinities with other populations. Okay. So that clearly shows there was admixture between ancestral South Indian and ancestral North Indians. That's the reason they are giving. I give the best example is look at one phenotype of skin color. Right? We have extreme light skin color. We go to north. For example, what you see here, the top one, which is closer to here, is the Kashmir Pandit. On the other hand, you have tribes. The skin color varies from extreme light to extreme dark. In between there are so many colors, all because of the admixture. That is again clear from this. There is a gradient of cluster. If you go to extreme north, you will find 90% of ancestral North Indian, 10% or 5% of ancestral South Indian. Similarly, if you go to extreme south, you will find 90% or 95% of ancestral South Indian, 5 or 10% of ancestral North Indian. So in between, there are so many percentage. Okay. And what we have concluded in that study was all Indians are admixed. Right? There's no doubt. And we found also high frequency of recessive disease as a possibility of because of the admixture and endogamy, which I am going to come in later. And we also refuted the Aryan invasion theory. There is no specific group in India called as Aryan, right? Because we are all admixed. And overall, if you see what happened so far is these two are founding populations, ancestral South Indian and ancestral North Indians, and each one gave rise to many population groups. And during the last 2 to 4,000 years, all the population in India are admixed. This color indicates between these two, and different proportion of color indicates different proportion of genome. After admixing, every single population started maintaining the endogamy. Endogamy means marrying within the group. You all know that very strict endogamy practice is in India, but exogamy is happening at very, very low level. So this is the one which every population follows. In addition to that, what I have shown here is very few studies which has very important conclusion. We also have looked at several other populations. As you see here, these are all populations, these are linguistic groups, religious group, and these are ancient DNA, and these are different population groups. These are population from outside India. Okay. So what I have shown is these two populations, Andamanese and this particular study. After all these studies, our conclusion about India regarding population. We have the very first out of Africa. Subsequently, there are several migrations, and recently there are several migrations, right? Including Jewish, Siddhi, Parsis, and so on. So all of these made Indian population very complex, and in fact they have also shown that population in India actually evolved from the ancestral tribal, then the people who have migrated are mixed and is grown to the upper caste, so-called upper caste in terms of social classification. That means all of us shows some kind of genetic affinities. So that makes India is one of the very, very complex nation in terms of genetics, social, culture, many other aspects. And you would wonder to know that in India, the present day India, we have 4,635 anthropologically well-defined population groups. Nowhere in the world we have such a huge human diversity that includes 532 tribes, 72 primitive tribes, and 36 are still practicing hunter and gatherer lifestyle. They just go and hunt for food in the jungle. They don't do any job including agriculture. Okay. That's the present day population. So now I will take you. So this is what the first part of my study. Who are we? Hope that you got some idea about who are we, right? The second aspect is that what is the impact of the 2000 years of endogamy, which I showed earlier. For the last 2000 years we are practicing endogamy. Maybe the caste system which arose or which must have put more pressure on every single population to strictly maintain the endogamy management. To understand the impact of the endogamy on our health particularly, we took another study. In this study we have included 275 populations and collected 2,800 individuals. This time we are not restricting the study only in India. We extended the study to other South Asian countries including Pakistan, Sri Lanka, Nepal, Bangladesh, and so on. So this study in fact gave us very, very important clue that there's a lot of opportunity for us to study every single population, to understand a population specific mutation that may lead to population specific recessive disease, right? As a student, you all have got more opportunity in this field, right? Just listen that how much you can explore from this study. This is just one technical slide. I put it in simple way. Every single line what you see, there are 275 lines, as I said, 275 populations, represent one population. Right? The length of this line suggests that scholars IBD score, identity by descent, more IBD score, that means that population have more number of individuals share a same haplotype. Haplotyping, for example, if you compare this side of the audience, maybe you are 50 compared to other side. Out of 50, there are 25 are very close related. There, out of 50, only 10 are close related. Here the IBD is more because more closer. When you compare, for example, 10 kb sequence, it is identical. That means those 10% or 50%, whoever having the same sequence, that means they all come from a single parent, a single common ancestor who have given rise to multiple generation. And today we have many individuals who are having a same haplotype. That means DNA sequence is identical. Based on that, you can see if the IBD length is more, that means they all share the common sequence or come from a single parent. But here, that means every single individuals are different, not much related. In this line, also included two different population groups. One is Ashkenaz Jewish, other one is Finnish population. These two populations are well known for their endogamy, and it has been identified that there are large number of diseases in Ashkenaz Jewish. These are genes, these are diseases, and you can see autosomal recessive is predominant there. You know that the autosomal disease can be classified into two, autosomal dominant and autosomal recessive. Right? We all have 46 chromosomes, or you can say 23 pairs. Out of 46, 23 we all inherit from the mother in the form of egg, 23 from the father in the form of sperm, and fertilize, then we will again have 46 chromosomes. If you take a particular chromosome, for example chromosome 1, and there is a mutation in one of the pairs, which probably might have inherited either from the father or mother, with the heterozygous condition, that means one of the pairs having the mutation, some individuals have the disease, it is a dominant. But in the one of the pairs has a mutation, other one is not having, and they don't show the disease, that is recessive. It requires one more copy to express the disease. So here, what you see in Ashkenazi Jewish, most of the diseases are autosomal recessive. I mean the mutation must have arose sometime back in the population, and it is circulated because they are marrying within the community, and when there is an opportunity, the two heterozygous individuals marry and they pass on the mutation to the next generation. Both the chromosomes which has a mutation is present in an individual because the disease is a recessive disease. Right? Same way is in the Finnish population. Then you can imagine with this tiny IBD itself there are so many diseases has been mapped. Look at so many Indian population which has multiple fold of IBD. That means we don't have any clue that what kind of disease exists in most of the Indian population. To give you little more about the recessive disease, look at this pedigree. I hope students all know about the pedigree, right? If you put circle, that means female, square, male, and put a line, that means these two are married. Put a line down and put the circle, that means daughter. Right? Put a square, this is a son. Here the pair of chromosome, this chromosome has a mutation, this is not having the mutation, that means heterozygous. The individual is absolutely fine, there is no disease. Her husband doesn't have that mutation, it's a wild type, it's a heterozygous. Then they married, two daughters, again the mother transmitted that mutation to both the daughters, and both of them married somebody from outside, they don't have that mutation. So no problem as long as it goes in a heterozygous condition. Same way this goes in the heterozygous condition. So now, these two individuals, now because of so many generations has passed, they may not have any idea that they are related. Right? In a small village you go, there are some 50 families or 100 families. All the 100 must have come from a common ancestor, one grandparents. But today they say that, okay, they are unrelated, but they are in the same village. So that's the situation here. The girl and the boy received the mutation which comes generation after generation, heterozygous condition, both of them. Now they want to marry, both have heterozygous mutation, they are absolutely fine. But when they marry, pass on this chromosome, there's a statistically 25% that they can pass the abnormal chromosome mutation. Only then that has a disease. Okay. Now I'll show you the actual example. This is one of the studies where there's a mitochondrial disease which was caused by mutation in the Pol gene. In fact, we started the mitochondrial disease when Madurai was there, and he was the one who wanted to do mitochondrial diseases for his PhD work. Right? So there, see, this is the polymerase gamma nuclear gene, which is essential for mitochondrial DNA replication. You know that for DNA replication you have the polymerase, right? This is polymerase gamma, which is essential for replication of mitochondrial DNA. This is a wild type. There is a single base mutation from C to T. The parents, both of them are carrying mutation in heterozygous condition. You can see C and T are overlapping the same position. That means one chromosome is having C, other chromosome is having T. So heterozygous individuals are absolutely fine, 45 years and 39 years. But unfortunately, 2 out of 3 children are with homozygous mutation, like what you see here. Okay, so therefore, it leads to abnormality in the mitochondrial DNA replication. This is a wild type where you see intact around 10 kb band. Because of the mutation, there is a multiple deletion. Instead of here, there are multiple bands that show that multiple mitochondrial DNA deletion, which leads to several abnormalities. The brain has lesions, along with that encephalopathy, seizures, stroke-like episode, and most importantly, the children are dying at very, very early age. This is one example that you can see. This is the consanguineous family, this is a non-consanguineous family. Again, you can see a single base mutation in a gene can cause so much problem. That's because of the environment. So now I'm coming back to the slide. So until we did this work, we don't have any clue whether these populations, which has a very high IBD score, have any disease in their population or not. So we went back, we looked at some of these, selected a few populations from South India, went back, and one particular population near Madurai, we found there is a disease called junctional Herlitz epidermolysis bullosa. And when we looked at the child, has skin rashes all over the body, including congenital heart disease, and the children dies very, very early age, within 2 months after the birth. Then we did exome sequencing, that means sequencing all the genes, and found a mutation, 11 base pair deletion from here to here. This is a wild type and this is a deletion. In a gene called LAMB3. So that is responsible for that. But until, I'll come a bit later, but this is what we found. So probably the 11 base pair deletion in this particular gene may be responsible for the disease. So far no report on this. Then we looked at the sequence which we had, few thousand. Then very recently we completed sequencing 10,000 whole genome. Very recently, January, Honorable Prime Minister announced about the completion of the 10,000 genome. We looked at in all the 10,000 individuals. These are 10,000 individuals. You can see how diverse these populations are. There are 83 different population groups. There's an Indo-European here, there's a Dravidians here, Austroasiatic in this cluster, Tibeto-Burman in this cluster. Again, none of the individuals carry that mutation. Then we looked at the global database. These are the global database, which consists of 8,62,000 individuals. We look at the chromosome, 17 lakh chromosome worldwide. We found only 2 individuals from South Asia carry the mutation in heterozygous condition. One chromosome carry the 11 base per deletion, other one is normal. Okay. So it's very, very rare mutation. This is for the first time we are showing this. But when we are identifying the first time, always doubt whether this is the real cause for the disease or not. So what to do that? We have to generate animal model. First we looked at the sequence. This is a wild type sequence. This is where the mutation. This is the site where the mutation is. It produced 1172 amino acid, the complete protein. Because of the mutation, it truncates, thus introduce the termination codon. Therefore, it has reduced the protein size, the amino acid size is reduced to 799. I mean, 373 base pair amino acid is missing. Right? So we thought, let's generate the animal model. We precisely deleted using CRISPR-Cas9. I think you know the technique, right? CRISPR-Cas. It's a technology where you can delete even single base pair. So we precisely deleted this mouse strain, and we found the pups are actually developing the kind of rashes, scaling, but unfortunately they don't survive beyond 2 days. Right? So then, fortunately in this process, you also know that there's an off-target effect of CRISPR-Cas. We got another clone with 45 base pair deletion that consists of 11 base pair here, the 45 base pair. While the 11 base pair deletion leads to termination of the amino acid, which removes 373 amino acid. Whereas here, this is a non-frame sheet, removes only 15 amino acids. Therefore, the impact of those miles, which has the 45 base pair deletion, is much lesser. So up to 4 months, they are absolutely fine. Then slowly it started showing the phenotype. And after 4.5 months, you can clearly see that the ratios and similar phenotype what we have seen in the children. And the internal organs also you can see this edema and so on. And internally, when we dissect it out, shows there's a dilation of heart, enlargement of spleen, and stomach size is reduced. And using the micro CT, you can, this is a very small heart. This is a small CT scan, micro CT. We looked at the heart and found there's a dilation. In this case, these are wild type and heterozygous, where you can see there's a hypertrophy. The interventricular septum is very thick. There's also hold, the septal defect. Cross section also you can see. And both wild type and heterozygous are almost same, but whereas you can see there's a hypertrophy. When we looked at the skin, the wild type is very intact of both epidermis and dermis. In the mutant, you can see there's a gap between dermis and epidermis. This is where the blisters start coming. Okay. Then to understand the human, because human homozygous doesn't survive, right? The children die within 2 months after the birth. We created the iPSC from the amniotic fluid. mothers with the heterozygous. We removed the 11 base pair from the wild type, made the homozygous line and tested with all the markers, and this is where precisely we deleted the 11 base pair. Now we have the homozygous line of human, and we again tested with keratinocyte and keratinocyte markers. You can see this is a wild type. The skin is normal, where you can see the homozygous keratinocyte shows abnormal. Okay, and we are continuing this work doing transcriptomics, proteomics, and also looking at various other aspects to understand the basic biology of this mutation. On the other hand, we have to help the society because we found the mutation which is responsible, because it's very clear from the study that mutation is responsible for the disease, right? In the animal model shows. Then we came to know the mother, one of the mothers who have lost 2 children are pregnant. Then we took the mother to Chennai to collect the amniotic fluid, and we tested and found this is the amniotic fluid. It's homozygous, heterozygous due to allele. Similarly, the amniotic fluid also showed fortunately 2 bands suggesting the heterozygous. Again, we sequenced and found that it's heterozygous. Then we advised the mother to continue with the pregnancy, and the child was born a couple of years back with the normal phenotype, and she's absolutely fine. It was also second time another mother we found with the help of Vaniraj. We collected sample in mother itself, and that also fortunately heterozygous. So the baby is again fine. So now we can't stop here. Knowing that we identify the mutation which is responsible for the disease, we need to do something for the society. The population is not very big, 5,000 individuals. We could have gone and collected the sample, but we wanted to go systematically, so we approached the Directorate of Public Health and Preventive Medicine, Government of Tamil Nadu, to give us the permission. And there is several procedures, including the ethical committee clearance and scientific committee clearance and so on. So finally we got the approval in the month of April, although the letter was showing January, got April, that we can go and collect the sample and screen. And very recently we went and collected the sample in July. We collected 180 samples. Out of 180, 4.5% of the individuals are carrying the heterozygous mutation. Okay, they are absolutely fine. Right? So we now planning for going and collecting all the individuals. Once you collect the sample from all the individuals, we can make a database. Even the children, for example, we find the heterozygous mutation today, and the child grows maybe after 15 years or 20 years, and he or she want to marry somebody, and that record also will be there, and see what is heterozygous. We can give appropriate suggestion. And again, in somebody with already married who are pregnant, we can do the prenatal diagnosis. In those cases, we can give premarital counseling for those who wanted to marry. So that way, it will be very, very helpful. With these background, when I was in CDFT, knowing the problem of pediatric rare disease, we initiated a mission program on pediatric rare disease. There are 16 different institutes which are part of this program. I am still coordinating. What happened here is the laboratory which routinely do the genetic screening, they will do. If they find the known mutations are not existing and the phenotypes are very unique, then they send the sample to CDFT. CDFT will do either exome sequencing or whole genome sequencing. identify the mutation, then predict whether the mutation has any potential cause for the disease. If so, then we use different animal models. Once the animal model confirms, then appropriate counseling will do. And we also want to correct it using some of the techniques which CMC established recently for hematological disease. And most importantly, we want to give a kind of public awareness. We generated 8 video in 8 different languages, including Tamil. Somebody is interested, you can just type pediatric rare disease, you go into website, and there will be 8 videos which will give information in 8 different languages. And in case if you come across any rare disease, you can also refer them to CDM. So there is no cost required because the project itself has funded by DPT, which will take care of everything. There is no single page required. So this was inaugurated by the then Vice President, Sri Venkaiah Naidu. Then program was launched by Secretary DPT, Dr. Rajesh Gokhale. So that will take the next 5 years to get sample from different centers and keep doing it. And if you recollect the IBD score, in India we have at least 1/3 of the population are with pediatric rare disease or rare disease in general. So unless otherwise we do, nobody else from outside going to do because it's a problem of India. So we have to do as students, you have a lot more opportunity to go into this field and explore yourself. So I initially mentioned about Jansky. I also want to mention about one of his another title which he published in the journal called The American Biology Teacher, published in 1973. Nothing in biology makes sense except in the light of evolution. Right? You'll understand how important this statement is. How evolution, in my story, how evolution, studying evolutionary biology helps us in understanding our health and disease. And finally, I want to show another paper, because in this paper it says making evolutionary biology a basic science for medicine. If I zoom the abstract, it says our general conclusion is that evolutionary biology is a crucial basic science for medicine. They also say that evolutionary biology is not just another topic vying for inclusion in the curriculum. It is an essential foundation for a biological understanding of health and disease. I end by thanking to all those who have contributed this work. You can see Deepakan Karthik, the one who contributed the last part of the study. Again, you can see many more in my lab people, including Niranjan who is here, Dr. Singh, and there are several collaborators across the world also who contributed immensely. That of hospitals involved in that, and the funding has come from Genome India project, which I showed one slide on that. Pediatric rare disease, CSIR, UK India Education Research Initiative, and Department of Biotechnology, Department of Science and Technology, and Medical Research Council at London that just concluded that project. Thank you very much.
I have only one question with the professor. He has given a very good narrative on the who we are. It's a very good title, I should say. So we know now we are in the seminar hall. Okay. Now I have a question with the professor. He has studied the pediatric rare diseases. Whether that particular group is being extended up to what age they will be observed. if they survive, if they cooperate with your study.
Yeah, so mostly
Thank you.
pediatric rare disease, unfortunately, like the one which I showed, so they lose their life. ICMR now has rare disease program. There are several committees have been formed. Look at various, including neuromuscular disease and many other diseases. So those are all adult rare diseases. So now, at present, there's a good investment in terms of looking at the rare diseases.
Thank you for the wonderful presentation, Professor. And as a Homo sapien, do we really evolve? If the answer is yes, in what way we are evolving? If the answer is no, what prevents us from the evolution?
Yeah, it's a very interesting question. So we in fact evolving at micro level, right? So as you know, evolution takes place millions of years.
Yes, according to Darwin, it will take a very progressive step and it is very slow.
Correct. If you look at for the last 100 years, where industrial revolution, the evolutionary process is little faster. For example, I told you about what happened in the recent past about domestication of animals. After domestication, the lactate persistent gene has changed. After agriculture, amylase gene multiplied. Right? Same way, the adaptation gene. Somebody want to live in high altitude, they have to carry more oxygen in their RBC, so that mutation evolved. And because of malaria, we have mutation which causes sickle cell. Right? Same way in the recent past, the mutations are happening. We cannot consider that as a major evolution. This is a small evolution. Even the skin color. Look at Africans and people from Europe. Skin color mutation arose because people migrated from Africa to a place where there is no sunlight. The Europe, even today, some countries in weeks, months, there is no sunlight. Sunlight is essential for vitamin D synthesis. So because of that, there is a mutation introduced in our gene SLC25A. So that turned the skin color into lighter. So therefore, even with the mild light, because of the less the melanin content, that's sufficient for vitamin D synthesis. That's the way it's happening. But the major evolution, for example, the back mutation, it happens, but they may not survive, right? They may not be able to transmit their genome to the next generation. That's the end of it. So unless otherwise it has an added advantage, it's not going to go. So slowly, for example, even the diseases, including cardiovascular and diabetes. So the reason, one of the reasons for diabetes is this thrift hypothesis. In our forefathers, they don't have much food. They have very small quantity, which are not rich in other fat and other things. So our genome is adapted for slow metabolizing with existing food. Right now, we have so much of variety of food, particularly with a lot of calories, like sugar, fat, and many more. Therefore, our genome is not able to metabolize that food. Therefore, we have the disease. Maybe start eating the same day, there could be a mutation. We don't know when it will happen. So that may take care of it. So it is happening at micro level.
It's a microevolution. And the another question is, what will be the reason for that changes in humans? You show the pictures of some persons.
Which one?
The pictures that shown in
the child.
Yeah, not a child. That monkey-like thing.
Unfortunately, we could not get the DNA. In fact, about 30 years back, that is the time when I joined CCFP. So this family, these three brothers were in one of the circus company in Bangalore. So At that time, the cardiologist told me, why don't you go and take the sample. I went to Bangalore with the help of one retired IPS officer. We went there, but still the owner of the company did not allow us to take the sample. If we would have taken the sample, we would know what is the reason for that.
So, is there any possibility of getting atavism? Can we predict it as an atavistic effect?
No, that could be some back mutation which we gain because they are like chimpanzee-like feature, you know. From chimpanzee-like feature we gain because of mutation, the way we look like today. So there is a back mutation which happened there, and also in the family, Turkish family which are with 4, right? Because we gained mutation with the straight posture, right? So the back mutation. Unless otherwise we look at, I think that family is being sequenced by some group. I don't know whether they publish or not. That's very recent.
Thank you, sir.
Thank you, sir, for the wonderful presentation. Thank you for the detailed presentation. I just have a couple of questions.
Yeah, please.
Number one is, did you observe any deviation in cardiovascular equilibrium? And second,
Which one?
that disease which showed report.
For this,
Pediatric.
okay, the epidermolysis
Is there any deviation in cardiovascular equilibrium?
Sometimes we can't actually apply statistics because there's no control, there's no one else is there. Only this particular population is present.
So, is there any role or founder effect on the genetic?
Yes, it is a founder effect in fact. Because the mutation is present only in that particular population.
Thanks.
It's very interesting to see the outcome of the You talked about one of the cases with the autosomal recessive disease. Have you encountered any autosomal dominant disease?
Autosomal dominant diseases are very common. Those who have mutation, they will show the disease. Because we don't want to actually look into that. Because obviously, whoever is having mutation will have the disease. So we wanted to see the hidden one. Right? Because it's amplifying in the particular population. Unless otherwise it exists homozygous can cause the disease. So when you go to the population, we found there's a 4.5 to 5% of them are carrying the mutation. This is what we wanted to contribute. How to eliminate this mutations.
Yeah, what would be your counseling suggestion for the autosomal dominant disease? It is not with endogamy.
But even with endogamy also you will see sometimes the mutation can happen at any time. So those who have the mutation, obviously the next generation might go. But in those cases, it will be the best one is pediatric diagnosis, right? To avoid the next generation, the fetus, to check, and then they have to take the decision whether to continue the pregnancy or terminate it.
Thank you.
Good afternoon, sir. I have a couple of questions about the male lineage and the female lineage differences in the ancestral North Indian and South Indian population. Because I heard and read about your story, your papers that they show differences in the male lineage by studying the Y chromosome. This is a different story, while the female lineage by studying the mitochondrial DNA tells a different story. So how they are related with the ancestral North Indian and ancestral South Indian population?
As I said, I didn't touch upon anything about the Y chromosome, which we all the males inherit, maternally, paternally. We had a lot of other stories because of the time. When you looked at the ancestral North Indian and ancestral South Indian, we had looked at the autosomes, which is equally contributed by both the parents. When we see the difference only the population where there is a male-mediated migration, for example, Siddhis. Siddhis are the ones who have been brought by Portuguese traders from Africa about 400 years back. So to work in the armies of Nawab and so on. Where more males have come. And you can clearly see, if you look at the Y chromosome mitochondria, they have more of Y chromosome, which are of Africans, and more of mitochondria, which is of Indians. That shows that the males have come and married Indian. So only those kind of male-mediated migration or female-mediated migration, you can see the difference. Whereas the admixture, which happens equally, you can see that both have contributed to each population.
So my next question will be about the availability of whole genome sequencing data. Is it free available or should we have some collaboration?
10,000 genome data is at the Indian Biological Data Center in RCD, one of the DBT institutes. DBT itself has called for a proposal to make use of that. So that's one way. The second way is directly somebody is interested in looking at that can apply. There's a committee which will look into that and give the permission. For that there's a lot of documentations required. At present it's not been released because the paper is being written. Once the paper is published, it will be available for all the researchers.
My last question will be on the DNA bills. You are one of the important members in that committee.
Which one?
The DNA bill, sir.
DNA bill.
Your thoughts on that, sir?
In fact, DNA bill was not long back by DBT and it has evolved multiple times. When I was a director of CDFD, there was a big COVID time. DBT called for a meeting because that bill has gone to Parliament. Parliament raised many questions and to address that question we have been invited. We again addressed those questions. One of the MPs from Andhra Pradesh, not Andhra Pradesh, from Telangana, whom I met later on, he has a lot of questions because of this bill. Some population may be discriminated and so on. So I explained him in person later on that for this bill, the DNA is concerned only the short tender repeats will be used. Those are used for identifying every single individual. That's a short tender repeats which is distributed all over the genome, used for forensic application to distinguish every single individual. Alleles are large number using 20 different short tender repeats. One can be able to distinguish whether this person belongs to this family or he is the father of the child or she is the mother of the child or this. It won't give other information about which population or which religions this person may belong to, which country. For that we have different markers. Right? I explained him, but he was not convinced, of course. But later on what happened after the bill went back to Parliament, then the Home Minister wanted, because that was in the scientific ministry, want to take that into Home Ministry. Then there's a lot of discussion and they also called us for the discussion. There was one data center under the Ministry of Home Affairs. They want to give SOP to all the forensic laboratory, how to generate the data and so on. That also went. Then again went back to Parliament and that's it. That's the end of it. It didn't go further.
Sir, you have made a wonderful presentation. What is your take-home message on endogamy?
I clearly showed that endogamy should be avoided, particularly when you have the recessive disease. I don't say that totally stop, but it always have some kind of there's possibility that there could be a recessive disease.
Should there be a bill passed for that, sir?
That we can't say.
Because the Indian community is very much on endogamy.
Yeah, yes. But on the other side, if you identify the mutation, that can be further taken at the population level, try to help the population avoid those mutations. That will be alright.
Are there any environmental factors?
There are for the mutation, it can happen. Randomly any mutation can happen. Yes. So this mutation also must have happened spontaneously. Some mutations are selected. There is a mutation which causes sickle cell and so on. That is selected for the particular environment. That is essential. Otherwise, random mutation, we all gain random mutation at different point of time. But the mutation, if it is present in only 1.2% of the genome, which is coding for the protein, if it is the rest of the place, it doesn't cause any phenotypic effect. It's a polymorphic. We all carry millions of variations, about 4 millions of variations. If you compare them. So those are not causing any disease. The mutation which is present in the coding region only cause the mutation, only cause the disease. That is in only maximum 2% of the genome.
Have you identified any environmental factors which can cause this mutation?
No, specifically we didn't look at it, but it's very well known. For a place where more radiation is there, it causes cancer. If somebody exposed to some kind of chemical radiation, these are all causes somatically. But it won't go to the next generation. Only the germline mutation goes to the next generation.
And one more last question, sir. Can these type of research be carried out in colleges?
Yeah, absolutely. Anywhere, whoever is interested, and only thing is funding is required. Otherwise, if you have any idea, most welcome to do that.
Will you be able to help us?
Definitely, one of the way I can help, I will be happy to help. Yes.
Thank you, sir. Thank you.
Yes, yes.
Our presentation was very impressive, and I'm totally from biochemistry department side, so I don't know much about this zoology, but you gave a brief explanation about the topic and who we are. And what is my question is that the autosomal recessive disease is what, like, in which category we can separate the disease, like based on the affected cell or based on the genetic markers, so on. So in which ways we can separate that autosomal disease that present in the individual?
Good, happy that one student asked. Sit down. So autosome, if you take the chromosome, I'll take the cell. We have nuclear chain. Where is the 46 chromosome? 44 are autosomes. Okay. One chromosome 22. There's one X and one Y. These are called as sex chromosomes. In the female, there will be 2 X chromosomes. In Y is present only in male, 1 X and Y chromosome. If it is on the Y chromosome, it's called as Y-linked disease. If it is on X chromosome, it's called as X-linked disease. Right? If it is on autosome, it's called as autosomal disease. If it is a dominant, one of the pair has a mutation cause the disease, it's called as autosomal dominant. But whereas both the copies of the chromosome required to cause the disease, autosomal recessive. Okay, these are the classifications. In addition to that, there's a mitochondria in the cell, right? Mitochondria has its own DNA. It's called as mitochondrial DNA. It's very small, tiny, 16,500 base pair. Only 37 genes are there. Of that, only 13 codes for protein. There are others are all tRNA genes. Mutation in mitochondrial DNA also can cause the disease, called as group of disorders called mitochondrial disorders, which always cause multiple system disorders. For example, one mutation which can cause a disease called melas. We expand that, mitochondrial encephalopathy, lactic acidosis, stroke-like episode. That's because it have multiple diseases. Another one is CPEO, chronic progressive external ophthalmoplegia, causes eye disease. Right? So mitochondrial DNA mutation can cause disease of all the organs. Okay, so that itself we can talk about 2-3 hours.
Thank you, sir.
Thank you so much for your excellent presentation. Any more questions from the UG students from this side? Self-listed genetics and evolution. இந்த semester யாரெல்லாம் pay பண்றது? அவங்க சாய்ஸ் விட்டுருவாங்க எவல்யூஷன்.
Thank you for your thoughtful presentation, sir. As you have mentioned that harmful mutations like LAMP3 gene leads to the junctional epidermolysis. So, how we can differentiate it is from a recently originated mutations or from our ancestor founder event? Like these harmful genetic mutations are from our own ancestor founder event or recently mutations. How can we differentiate that mutation, sir?
Yeah, so that's an interesting question. Yeah, you can say. So you can look at the population which has diverged very recently, close population, and based on the mutation and mutation existing in the flanking region, there's a statistical method to estimate the age of the population, age of the mutation. So based on the mutation, you can predict how many thousand years back or how many generation years back that mutation arose. For example, this mutation must be very recent because the same population is also living in other district, but they don't have that mutation. That means it is very recent event. Whereas on the other hand, I did show you that particular study. We found a mutation, 25 base pair deletion in myosin binding protein C3 gene, which causes cardiomyopathy. Okay. We sequenced the flanking region of the mutation. Then compare that because that is well spread across India and South Asia. Then we compare the gene with the chimpanzee. And based on the mutation existing there, based on the mutation existing here, we calculated that mutation arose about 33,000 years back. Right. So based on the mutation which are existing in the flanking region, you can be able to calculate the age of the mutation. Got it?
Any other questions? UG students?
So you, those who have not liked genetics and evolution earlier, did you now interest in genetics?
From Lakeview College. I'm firstly in my first year PG. That was an impressive presentation, sir. My basic question is, so the mutations, we all carry our ancestral genes, right, sir? So now the mutation is being occurred and we express the genetic disorder. Why is it not expressed in the ancestors back then itself?
No, because the mutation arose, whenever the mutation arose, from there, because it changes the amino acid and protein structure, because earlier it was not there. Gene is highly conserved, but the mutation which makes the difference, change the protein structure overall, because every protein is required for our function. If the protein is not complete, it is truncated, it may not be able to do its function. Therefore, the problem starts from that time onwards. The ancestor, there is no mutation, therefore they are perfect. For example, we are going in cycle or the same cycle is used. When there is no air in any one of the wheel, we can't do. No? The air is the one which creates the problem.
So should we conclude that the environmental that we are exposed to is actually the reason for the mutation, or?
Not always. So environmental mutation or sometimes for better to live in that environment. For example, the mutation which happen in the sickle cell mutation. So, in the malaria prevalent area, that mutation arose, therefore the RBC, which is round in shape, becomes sickle shape. Therefore, the malarial parasite could not enter there. Therefore, they are free from malaria, whereas they have other issues. Okay. Similarly, another example I showed, the lactate persistent gene, the skin color mutation, then there are several such mutations. In the high altitude, somebody goes, they may feel because of the less oxygen. Those who are in high altitude, they gain a mutation. Therefore, with the limited oxygen environment, the RBC can carry more oxygen. Therefore, they don't feel any problem. So that kind of mutation, which is essential for adapting in that particular region. Those are essential. But randomly, some mutation can happen when we are exposed to radiation or chemicals. So that is mostly somatic. Okay, it will not be passed on to the next generation. Okay. But those who are exposed, they may face many issues, including cancer and so on.
Thank you, participants. Thank you, sir, for the fascinating and thought-provoking lecture. Your insights on human...
Good afternoon, sir. I'm Prakshita from Zoology Radio College, and my question is on the prediction with future aspects. So, inherent violent behavior of humans lead to evolution. I do accept, sir. I have a doubt in, some people tend to change, or their biological clock gets changed due to some characteristic feature difference. Say lifestyle changes like sleep, etc. And if that changes occur, that is not manipulated mandatorily, but do it. So people will be getting involved.
Yeah, sit down. So what you are saying is that how the behavior or circadian rhythm, for example. So that is somebody practice for anything. We need a practice. For example, somebody is playing any game. So they need to have practice, including sleep. I sleep for maximum 4.5 hours. Sleep at 1 o'clock, 2 o'clock. I can get up at 6, 6:30. That I practice right from my PhD days. Right? If I sleep more than 5 hours, my body is not taking. So that even today, even last night, also I slept at 2 o'clock. So there's always some work to do. Manirajan knows that. Because after dinner only we discuss in the lab. Right? When it was the PhD. So at least now I am sleeping at 2 o'clock. Those days I sleep at 3 or 4 o'clock. So that is the habit. That became a habit. Right? So somebody again, if I want to go back to the normal, I can practice that also. I don't want to do that because there's a lot of work to do. So I practice that. So both practice and environmental influence is very important.
Yes, sir, but still people tell that do not change or do not disturb the biological clock. Even they start insisting on this. But at initial stage, if it is a disturbance, we'll be getting involved. About the distraction or disturbance, so it will not be there. Later we'll be getting adapted to. Am I right, sir?
Yeah, absolutely. Yes. Initially you may feel a little bit, but once you adopt, it will set tone for the future. No, there are for everything there's a genetic basis, right? Genetic basis. But something which continuous practice can overcome that also.
I read an article insisting on
created for the adaptive, to enhance the adaptive feature of disturbance of balance control. Is it fine to
No,
go with that?
sometimes because to control some of them, they are very hyperactive. For example, there are drugs which control their hyperactivity. That is considered as a medical treatment because we cannot leave because naturally they are hyperactive. Right? They have to be controlled. So in that case, yes, there is. Yeah. And also, one example I can give, there are some population which has a mutation which lead to even death if they are treated with certain drug. For example, one population called Vaishya. It's a very small community. They have mutation in pseudocholinesterase gene, PCH gene. Along when somebody is going for surgery, they gave anesthesia. They also give muscle reluctant drug. If those individual has a mutation in the PCH1 gene, if the regular anesthesia is given to them, they won't be able to metabolize it. For every aspect there is a gene. Whatever the drug we consume, there is a gene which metabolizes the drug. For example, 2 hours surgery, they give the dose so that for the 2 hours they will be in anesthetic effect. But after that, the gene metabolizes, breaks down, therefore they will come out. But Vaishya population, they will remain in anesthetic effect because of the mutation. They are unable to metabolize the drug. Sometimes leads to paralysis, sometimes leads to death also. So everything, there is a genetic basis.
Thank you, sir, for your wonderful and inspirational expressions connected with I have a few questions. In 1 minute. For example, vitiligo, leukoderma, and a lot of medical diseases have been drastically increased nowadays. I think somewhere we are mixing or connecting evolution and genetics and genetic disorders and medical disorders. If you see the records of Keeladi or Adichanallu's excavation and other records, that will tell more about our ancient people, lifestyle and culture. We disobey to the nature nowadays more than animals. We violate a lot of the laws in the nature, disobey to the God. Therefore, more number of medical hospitals than the veterinary. Yes, sir? Therefore, we are I think mixing the genetics and evolution with a lot of the current scenario of the disease enhancement.
No, we are not mixing with the genetics and evolution. That remains the same. But what is happening environmentally in the sense, not only the environment in which we live, also the environment, food is one environment. The drug is another environment. So the way we travel is another environment. Because traveling to another place also one has to adapt to the environment. So there are many other issues when compared to the previous days. And also the selection which happens. For example, somebody who is living in a remote jungle, okay, they are adapted to live in that particular environment, adapted to eat food, whatever they exist in the environment, they are happy. But now in the city, it's not like that. If you bring the people who are in that particular environment and bring it here, the environment in which we live, a lot of pollution, many pathogens and so on, they may not even survive because they don't have the ability to overcome this environment. For example, people living in Andaman Island, they are happy there. If you bring here, immediately they will have some kind of. So we are the one who created this kind of environment, right? and the food habit also added to that. That's why we have so much disease. And another issue which I want to tell you is, in this particular environment, those who have defective genes, for example, somebody has heart failure at very early age. In the city, we take the individual to hospital, make whatever the necessity to treat the individual to survive. So what happens? The person survives, marries, he or she, and transmits the gene to the next generation, goes. Whereas in the environment where there is no modern medicine available, somebody has suddenly gained the mutation as a heart failure, that's the end of that person. Right? The mutation is not transmitted to the next generation. So whosoever is living there, they are all selected to live in that environment. But here with the modern facility, with the gained mutation, we are protecting that, and therefore the mutations are transmitted to the next generation. So that's one way one has to look at it.
Thank you, sir, for the fascinating and thought-provoking lecture. Now, to conclude this memorable session, I invite Dr. P. Velladurai, UG Head and Associate Professor, Department of Zoology, to propose the vote of thanks.
We would like to thank Dr. Paul Jaker, the Principal Secretary of this great college, for helping us and motivating us to conduct this type of program in this department. We would like to thank Dr. Dr. Michael, the former head of the Department of Zoology, for connecting the alumni and the present set of the zoology students of this college and other neighboring college, for sharing or connecting the subject with the zoology, especially this evolution, connecting with Dr. JCBA Endowment. And we strongly believe, sir, that this type of lecture, endowment lecture series, connecting the present set of zoology students with the former alumni and their good hearts for collecting or giving more funds for arranging this type of programs, for enriching the subject knowledge. We thank you so much, Dr. Michael, sir, for we strongly believe that God will give you more help and more spirit of understanding with your alma mater. Thirdly, we would like to thank Dr. Thangaraj, the speaker of this event. Shall you give a big clap to more than 1 hour lecture to this title, A Tale Behind Tracing Our Roots and Beyond. Hope everybody is enjoying the topic, because that reflects your questions. So many people asked several questions, and sir expressed or interacted with all such questions. Thank you so much, sir, for your coming straight away from CCMB Hyderabad, consented, accepted the invitation, and traveling a long time and visiting our college second time or the third time. Okay, thank you so much for your coming. We would like to thank Dr. Anil Selvam, Vice Principal of our college, for his kind-hearted help and everything. Dr. Beulah, the bossa, the financial officer of our college, for giving the college vehicles for transportation and financial assistance. We would like to thank Mr. Prince, the senior master Sainik School. Still, we look the same. I think physics and the spirit of understanding, coming back to the same B1 hall, recollecting all your good old days. Thank you so much, sir, for remembering your teacher and reconnecting with the present set of students. We'd like to thank Dr. Maripan, the former scientist, ICMR, and the former association president of PG Zoology Association, connecting our departmental activities every time. We would like to thank Dr. Joseph Tades, the former head of the Department of Zoology, for coming
to glorify the whole occasion, connecting with all people. We'd like to thank Dr. Nitya, the head Department of Biotechnology of our college. We'd like to thank Dr. Vannirajan, the scientist, Aravind Eye Hospital, the former student of the American College, and the CCMP. We sincerely thank you for coming and interacting with our college and the department frequently. Thank you so much. We'd like to thank all alumni, invited professors, friends, joined in the online. Thank you so much for all your presence and understanding this subject with Dr. Thangaraj, an eminent Bhatnagar fellow. We would like to thank Dr. Pavanraj, Assistant Professor, Department of Zoology, Vivekananda College, Tiruvadagam, for accepting our invitation to come and listen to the lecture, sir. We would like to thank all our own faculty friends. You are worked under a different committee to glorify this talk or this lecture and make this lecture series a greater success. I would like to thank all PhD research scholars, BSc and MSc zoology students of our college, and zoology students and biochemistry, biotechnology students of other neighboring colleges and the departments. Thank you so much, one and all, for all your patient listening and understanding this subject. You can pursue little bit of this take-home message, and you can go and do more research in the days to come, because more funding is available for this type of areas. If you want to do this type of research in anthropology, right, in tribal community or unattended people and their conditions, definitely you can come up in your life. So a lot of research and fundings are available for anthropological and other areas, right? Thank you so much for coming to this lecture series, and you can understand more in the days to come. You can connect with sir, Dr. Thangaraj sir, through mail and or through his WhatsApp number or mobile number for having clarifying your further doubts in the days to come. Thank you so much for all your presence.